Life, as you well know, is an energy-intensive process. It takes energy to operate muscles, extract wastes, make new cells, let the brain process and send messages, and even, from time to time, do some serious thinking. An organism's entire energy budget is spent on the cell level. In some cells, as much as half of a cell's energy output is used to transfer molecules across the cell membrane, a process called active transport. Cell movements require energy, and thousands of energy-hungry chemical reactions go on in every living cell, every second, every day. The kind of energy cells use is chemical bond energy, the shared electrons that hold atoms together in molecules. Most cell processes use the same energy source, the rechargeable energy carrier, adenosine triphosphate. ATP has this arrangement, a molecular unit of adenosine coupled to a chain of 3 phosphate groups, thus the name, adenosine triphosphate. The phosphate groups are held to each other by very high energy chemical bonds. But under certain conditions, the end phosphate can break away, and the energy released used for energy-hungry reactions that keep a cell alive. When the end phosphate is released, what is left is ADP, adenosine diphosphate. This change from tri to di is taking place constantly as ATPs circulate through cells. The recharging of ADP to ATP requires a huge energy investment, and that energy comes from the food we eat. How energy is extracted from food molecules and used to synthesize ATP is one of the great discoveries of modern biology. All nucleated cells contain mitochondria, the tiny bodies where ATP is produced. This energetic protist is literally filled with them. But the best way to see them clearly is to squeeze the cell under a cover glass until it ruptures, forming a thin membrane bubble, a little aquarium in which float a few of its mitochondria. A mitochondrion consists of 2 sacs made of membrane. Folds in the inner sac increase the surface area for chemical reactions that produce ATP. By breaking up mitochondria and separating out the membranes, biochemists have discovered exactly where the chemical reactions involved in synthesizing ATP actually occur. First, mitochondria take in molecules derived from food, molecules with lots of chemical bond energy, the breakdown products of sugars and fats. Sugar contains enough chemical bond energy to burn with a hot blue flame. Fat contains even more. Fat has about twice the energy content of sugar. If you can boil 1 test tube of water on a spoonful of sugar, a spoonful of fat will boil too. To understand how energy is extracted from these fuel molecules, we need to climb right inside of a mitochondria. In the space between the 2 sacs, fuel molecules are disassembled in a way that releases their chemical bond energy. This energy, in the form of electrons, drives molecular pumps embedded in the inner membrane. The pumps push hydrogen ions obtained from the fuel molecules into the inner membrane sac. It's like blowing up a balloon. It's during this process that oxygen plays its role. Oxygen has a powerful attraction for electrons. Think of the electrons released from fuel molecules as a stream of water. What oxygen does is lower the stream bed dramatically so that the water, the electrons, can do a lot more work. Some bacteria can live in oxygen-free environments, but also have the ability to use oxygen if it is available. Without oxygen, the cell can make 2 ATP molecules for every sugar molecule metabolized. With oxygen, the same cell can make 36 ATPs from each sugar molecule. Oxygen's powerful pull on electrons allows most of the energy in their fuel molecules to be used to push in the